Bismillah Rahman Rahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about Traptophane or Traptophane operon. In the previous video we talked about lactose operon and we talked that how it gets switched on and switched off. We talked that it will be switched on and switched off by this lactose and glucose, and we also talked that it will be switched on and switched off by this cap and cap complex. Now how, we already talked and explained it. If you missed this video lecture, so go back to my channel and watch this. Today in this video we are going to talk about Traptophane operon or Traptophane. Remember guys, in the previous video we talked that lactose operon is actually giving the positive signal. It is the positive transcriptional process, regulatory transcriptional process. Actually this is the video lecture which is related to regulation of transcription. Okay, and this is actually positive regulation of the transcription process, positive control of transcription. And the Traptophane is actually the negative transcription. It is the negative transcription. Okay, why? Because we talked that this Operon is normally switched off, but the Traptophane operon is normally switched on. It not need to be switched off. It normally switched on. So both are actually opposite to each other. That's why this is transcriptional. This is positive transcriptional. Means that this is switched on. This is actually giving positive signal, and the Traptophane giving negative signal. Now, how the Traptophane operon will be switched on and switched off? What's actually going on in Traptophane operon, and what is the structure of Traptophane operon? So let's talk about that. Let me clean the whiteboard and let's start. Okay friends, so now let me write, okay, or we call it Traptophane operon. First of all, we need to know that what is actually tryptophan is. Tryptophan is actually, it is amino acid. It is the amino acid which are specially used for normal growth of the cell. Remember guys, in prokaryotic cell the tryptophan are produced, but in eukaryotic cell tryptophan are not produced. We take the tryptophan from the diet, and it is responsible for the normal cell growth. Okay, now how this tryptophan is switched on and switched off? It is normally, it is switched on normally. Why? Because most of the time the cell need tryptophan for normal growth. The cell need tryptophan for normal growth, so it is switched on. Okay, so let's explain the whole process with the diagram. And it actually giving a negative signal. What it giving? Negative signal to transcriptional control. It is actually the negative signal of the transcriptional control. So this is actually the structure of Traptophane operon. It contains regulatory genes. As you can see, these are what? These are the regulatory genes, R genes, and these are the trap operon or trap genes. Let's suppose the trap genes consist of regulatory genes as well as the structural genes. And the this whole is actually, we also mentioned it in the trap operon system as well. Now normally it is switched on. So what happened if it is switched on? This operon contain promoter, operator and ladder sequences. This is very important. Okay, inshallah we will explain the ladder sequences in detail in the next video lecture, in upcoming videos lecture, not in the next video, in upcoming video lecture, because in the next video we will explain arabinose operon. These ladder sequences are very important because these ladder sequences also perform the switched on and switched off process, but it is not perform the switched on and switched off process in at that level. It will perform the switched on and switched off process at the translational level. That's why whenever we are explaining the regulation of translation, we will explain that ladder sequences at that video lecture, but not here. Now what happened? It contains ladder sequences. It contains these 5 genes A, B, C, D, E. Okay. So again, it is what this is, the polycistronic gene expression, and it will give us RNA. Let's suppose this is what this is, RNA, which we called it mRNA, and we called it polycistronic mRNA. So this is what this is, again, polycistronic mRNA. And from this polycistronic mRNA, what we get different types of the protein, and these proteins are, let me write, these two, as you can see, ED, it will produce, let me write here, this E and D genes will produce a protein we call that enthranylate synthetase. We have C. So this C gene produce enthranylate isomerase. We have B, which produce tryptophan synthetase beta. And then we have A genes, which produce tryptophan synthetase alpha. Okay, alpha. So these are the enzymes which are produced by these genes present in the tryptophane operon. All these enzymes, as you can see, are used for tryptophan production. Okay. So normally it is switched on, and all these enzymes are producing, and it will give us tryptophan. Okay, so this is switched on. If the cell need 200 tryptophan, let's suppose the cell need 200 tryptophan, so it is producing 200 tryptophan. But if tryptophan amount is increased like 210 tryptophan, if the cell made, so then it is creating a problem. This tryptophan operon should be turned off. Now how it is turned off? This is actually normally turned on, but if the production is enough, so then it should be turned off. So it will be turned off. What happened at that time? These genes will be activated. Okay, these genes will get activated. So remember, guys, when these genes get activated, it give us an protein which we call that. This is a protein we call that a repressor. Repressor or inhibitor? Repressor. But this repressor protein is apoenzyme, not in an active form. This repressor protein, we have tryptophan. Tryptophan is actually bind here, and then it is converted into holoenzyme or active form of the enzyme. So when we have tryptophan and repressor, the complex is formed, and it will sit to this operator region. It will sit to this operator region. So when will sit to this operator region, so then what happened? RNA polymerase, although RNA polymerase binded here, let's suppose RNA polymerase binded here, but still it is not moving at that direction. If it is not moving, so then what happened? The process will be stopped. No more tryptophan will be produced. Means tryptophan genes will be transcribed, and these protein are not produced, and the process are switched off. Okay, so this is how the process is switched off.